What does it take to build a cell? If we wanted to build one from scratch, how would we do it? Is it even technologically possible for us to make artificial life? These are exactly the kind of questions that researchers were trying to answer when they made Mycoplasma laboratorium, the world's first synthetic organism. Today, we'll be taking a deep dive into this microbe that was engineered to have the smallest genome of any living organism ever known. We'll have a look at which genes are essential, which ones are nice to have, and which ones you could toss out if you wanted to. Well, Mycoplasma could. And we'll also ask the question, is Mycoplasma laboratorium really artificial life, or is it something else? So the first thing to discuss is, why would anyone want to do this in the first place? Right? Aside from it being cool and to prove that we can. Well, one of the main motivations behind research like Mycoplasma laboratorium is to better understand just what it is that makes a cell a cell. See, modern organisms have been evolving for billions of years to become what they are today. And over that time, they've built systems on systems on systems that are all integrated with one another. This makes it difficult to understand whether a system is truly essential or whether it's simply so integrated into the cell that too many other systems rely on it to remove it from a modern organism. Think of it like a tower of cards. The first layer of the tower is what's truly essential for the cell to be a cell. But over time, layers are added. And as we get to layers 3, 4, 5, 6, now, if we were to remove a card from layer 2, the whole tower would collapse, even though layer 2 is not technically essential for a cell to be a cell. Even something that looks as simple as a bacterium has in fact been evolving for billions of years and is far more complex than the original cells that evolved could ever have dreamt of being. So for researchers who are looking to try and answer these fundamental questions about what exactly makes a cell, things like Mycoplasma laboratorium are essential in order for them to assess how truly required specific systems are. But of course, another main motivation was simply to prove that we could. See, before this work was done, no one had ever been able to engineer a genome in the lab and then implant that genome into a living cell and have it remain viable. Proving that we were capable of doing this would mean that as long as we can iron out several other issues involved with this kind of thing, that we are actually technically capable of creating synthetic life by engineering a genome that we designed from scratch in the lab and putting it into a cell as a carrier. These are really the two main motivations behind the work driven by scientists at the Craig Venter Institute that prompted them to spend years engineering Mycoplasma laboratorium. But how exactly did they do it? One of the first barriers to this work is that you need to take a DNA fragment the size of an entire genome and somehow get that into a living cell. And if that sounds simple, it's really not. Usually when researchers do genetics experiments where we introduce foreign DNA to a cell, we work with DNA fragments that are orders of magnitude smaller than entire genomes. And the success rate decreases substantially with the size of the DNA fragment. So the first thing the team did to test whether it was even possible to do something like this was to take cells of Mycoplasma mycoides and extract the entire genome intact in one fragment. Now, to get DNA into a cell, there are really three main techniques that we use: transformation, conjugation, and transduction. Of these three, transformation is really the only one that's viable for working with really large DNA fragments. The way transformation works is that the cells that you want to put the DNA into are taken and treated in a way that makes them more likely to take up DNA from outside the cell. There are several different ways that we can accomplish this. For example, electroporation, which is a type of electric shock treatment that we expose the cells to. We can also heat shock them, essentially moving them back and forth between freezers and heating blocks in order to shock the cells. We can also chemically treat them and dissolve outer layers of the cell, leaving the cell itself still intact, which reduces the number of barriers that the DNA has to cross to get into the cell. Or, in some cases, you can starve the cells, which causes them to intentionally take up more DNA in order to supplement their own metabolisms and help them survive. Cells that have been treated like this are what we refer to as competent. These cells are then taken and mixed with the DNA of interest that you're trying to transform them with, and several other compounds that promote DNA uptake by the cell, such as polyethylene glycol. In this case, the team took cells of Mycoplasma capricolum and made them competent by starving them. They then took these cells and they mixed them with the intact genomes that they'd extracted from Mycoplasma mycoides and polyethylene glycol in order to transform them with the genomes. Afterwards, they subjected these cells to selection, thereby removing any cells with the original genome from the mix. At the end of all this, the team was successful and were left with cells that had originally been Mycoplasma capricolum, but were now genetically Mycoplasma mycoides. So now the team had proven that they could take a genome from outside the cell and put it in successfully. They needed to prove that they could synthetically build a genome and get that genome into a cell. And to do this, they synthesized from scratch the entire genome of Mycoplasma mycoides in the lab and transformed Mycoplasma capricolum the same way they had done with the original genomes. Synthesizing the artificial genome of Mycoplasma mycoides was a long and arduous process that involved synthesizing small fragments of the genome, cloning these fragments together, replicating them, and then cloning them again together, iteratively, to build up the entire genome slowly over multiple rounds. There were some slight differences between the final genome content of the synthetic Mycoplasma genome versus the original, but in all the ways that mattered, the genomes were effectively identical. And in 2010, the team proved that they could transplant this synthetically engineered DNA, the whole genome, built in the lab, into Mycoplasma capricolum, and it survived and proliferated, making it, arguably, the world's first synthetic organism. As part of this, the team hid 4 watermarks in the synthetically engineered genome using a DNA-based code that they invented. The first one gave a link to a web page, which then gave instructions on how the decoder could contact the team to prove that they had solved the riddle. The other 3 were quotes, one by James Joyce, one by Robert Oppenheimer, and one by Richard Feynman. It's estimated that the synthesis of this genome costs overall around $40 million US, and the equivalent of 200 years of joint work by the researchers involved in synthesizing it. So now the team had proven that they were capable of synthesizing a genome from scratch in the lab and transplanting that successfully into a living cell and having that cell survive as long as the genome encoded everything that was necessary for the cell. The next thing they had to do was work out what is essential for the cell. And actually doing this is a fair bit easier than the previous 2 examples, because a very common technique in microbiology is called transposon mutagenesis. This is a process by which genes within an organism are randomly interrupted by a mobile genetic element called a transposon. Transposons are naturally occurring, and they basically distribute themselves throughout genomes as a selfish element. Most transposons display some kind of specificity or bias in where they want to insert, so they don't insert completely randomly. But several of them have been engineered for biotech purposes to act virtually randomly and can be used in order to knock out practically every gene in the entire organism and see whether or not it really is essential. The way that this works in practice is that the cells are transformed with something carrying the transposon. And the transposon is under the control of some regulator that requires presence of a specific compound, usually a sugar. So the cells are then put into media that contains that sugar, which activates the transposon, and it starts copying itself and inserting randomly into the genome. The cells are left in this state for a little while to allow the transposon to copy as much as possible. before being placed in regular media and growing up. Now, if a gene is disrupted that's essential, the cells where the transposon has inserted into that gene will die. If a gene is non-essential, those cells will survive. And so the organisms are then allowed to regrow, and after they've regrown sufficiently, they're sequenced, and the locations where the transposons can be detected are mapped along the genome. Any locations that don't show transposon presence must be essential. Any genes that do have transposons inserted must be non-essential. And in this way, we can quite quickly and effectively work out whether or not genes are likely to be essential for the organism as a whole. Based on a combination of transposon mutagenesis and some trial and error experiments, the team concluded that Mycoplasma mycoides encodes approximately 473 genes that are absolutely essential. Many of these genes are involved in processes that we would expect to be essential. For example, the genes involved in copying the DNA of the organism, in expressing genes, and in carrying out cell division were all virtually untouched by the transposon. Alongside this, the machinery required for the cell to make and maintain its membrane were virtually untouched, which makes sense because a cell without a membrane is not really anything. A significant number of genes involved in metabolism are also untouched. Again, this makes sense because without the ability to generate energy for the cell and to synthesize molecules that it can't get from outside the cell, no cell is really going to survive for very long at all, is it? Perhaps most interesting of all, though, is that 149 of the genes had no known function. We had no idea what they were doing and why they were apparently essential for the cell. The fact that such a large number of genes with no known function was apparently essential was a shock. This is not what was expected. In the time since the publication, there have been concerted efforts to try and identify the function of these genes in order to understand exactly what it is that makes them essential. And that number has been shrunk to 91. But that's still a lot of apparently essential genes that we don't know what they're doing. So, now the team knows that they can synthesize the genome from scratch, they can get it into a host organism successfully, and they know which genes they need to include to create a minimal genome. That's exactly what they did. They took those 473 genes, they synthetically engineered a genome that had only those genes, and they transplanted them into mycoplasma cells to create Mycoplasma laboratorium, the world's first highly reduced synthetic organism. Creating synthetic life is always going to be a controversial topic. Just because we can do something doesn't mean we should. And as soon as this project was announced, several bioethics organizations voiced concerns over it. The Canadian ETC group said that it was a chassis on which you could build almost anything. It could be a contribution to humanity through discovery of new drugs, or it could be a threat through development of bioweapons. This prompted Craig Venter, who was leading the project, to say that when dealing with scales of this level, you can't expect to keep everyone happy. Venter is known as a bit of a showman, and much of the media attention on this project was focused more on his grandiose statements than it was on the actual science, prompting Jay Kiesling, a well-known synthetic biologist, to say that the only regulation that was needed was on his colleague's mouth. Regardless of the question of whether we should do this, Mycoplasma laboratorium is a huge step along the path to proving that we can. But does taking an existing genome and deleting genes from it and then putting that into a new organism really count as being synthetic life? You could achieve the exact same end result by sequentially deleting these genes from the original organism in which they existed, and no one would consider referring to this as a synthetic life form. Does synthesizing the genome in the lab itself really change this? Mycoplasma laboratorium is a top-down approach to the development of synthetic life. Let's take an existing genome and remove genes from it until we get to the smallest one possible. Whether we can go in the other direction and add genes from nothing, building up a genome from scratch, is an open question and is the subject of several major US and European grants right now. The reality, though, is that we're still a long way from any of this coming to fruition. And for now, the applications that have been proposed for these synthetic organisms, such as replacing natural organisms that are used in industrial processes, are still unrealistic, because the synthetic ones are nowhere near as productive as the naturally evolved species that we're already using. Evolution remains king, for now. One thing that Mycoplasma laboratorium has given us, though, is a great deal of insight into just what makes up the fundamental building blocks of cellular life. Our understanding of how the first cells might have evolved has been aided quite substantially by the findings of what's essential for Mycoplasma species in the development of Mycoplasma laboratorium. Though there are obviously large missing pieces of information, as the remaining 91 functionally unknown genes that are essential show us. So, is Mycoplasma laboratorium really a synthetic life form or not? Let me know what you think down in the comments. If you like this video, consider hitting the like button and hitting the subscribe button and the notification bell if you want to see more content like this. And I'll see you next time.